Welcome to Immunopolis Central Laboratory, our high-tech facility for assessing the body's defense system. We are here not just to observe, but to rigorously test the functionality of our immune defenders. Our comprehensive evaluation process systematically assesses three critical parameters: the number of immune cells, their precise identity, and, most importantly, their functional activity. Our first stop is the immune census, the complete blood count with differential. This automated scan performs a full head count of the immune army, measuring the strength of its circulating forces. One by one, the machine tallies the key defenders: neutrophils, the rapid responders; lymphocytes, the strategists; monocytes, the reinforcements; eosinophils, the parasite fighters; and finally basophils, the alarm signalers. With these precise numbers in hand, we gain our first snapshot of immune readiness, a foundational measure of the immune system's overall capacity to defend. Next, we arrive at the identity checkpoint, flow cytometry. Here, immune cells line up and pass through the analyzer one by one, each under an intense laser beam. Using fluorescently labeled antibodies, the system scans the molecular badges displayed on every cell surface. As the technician asks, "Which markers do you express?" the cells answer in bursts of color. CD markers light up, revealing each cell's true identity and rank within the immune army. This precise interrogation allows us to distinguish T cells from B cells, separate helper from cytotoxic T cells, identify naive and memory populations, and uncover other specialized subsets hidden within the crowd. At this checkpoint, numbers take on meaning, transforming a simple head count into a detailed map of immune specialization and readiness. The flow cytometry data is visualized in a dot plot, which we call Quadrant City. Each dot represents a single cell, and its position within the four quadrants tells us which two markers it carries. This visual interpretation is essential for distinguishing key subsets, such as CD4 helper T cells and CD8 cytotoxic T cells, based on whether they are double negative, single positive, or double positive for the markers being tested. Moving beyond cells, we assess the humoral component through serum analysis. We quantify the key immunoglobulins G, M, A, and E, as well as complement components like C3 and C4. Techniques such as enzyme-linked immunosorbent assay and immunoelectrophoresis are used, in which the signal intensity directly correlates with the concentration of these critical defense molecules. Now we test the function, beginning with the lymphocytes at the training camp. Numbers and identity are not enough, a true defender must respond. In this phase, lymphocytes are challenged with activating signals that mimic antigen encounters, testing whether they can awaken from rest and enter rapid division. A healthy B cell answers the call by entering rapid DNA synthesis and proliferation, preparing for differentiation into antibody-secreting plasma cells and memory B cells. One classic way to measure this response is the thymidine incorporation assay using nonspecific mitogens, such as bacterial lipopolysaccharide. Here, radiolabeled tritiated thymidine is incorporated into newly synthesized DNA during the S phase of the cell cycle. The more thymidine taken up, the more intense the signal, a direct readout of how actively the B cells are dividing. Modern approaches complement this classic method. Carboxyfluorescein succinimidal ester, or CFSE, is a fluorescent dye used to track cell division, while Ki67 expression, a protein marker indicating cell proliferation, confirms entry into the active cell cycle. Together, these assays verify that B cells are not only present and properly identified but also fully trained and ready to expand and produce antibodies when real antigenic threats arise. In addition to the non-specific immune response, B cells are tested for their ability to respond to specific vaccines by measuring antigen-specific antibody production before and after immunization. Prior to vaccination, antibody titers are typically low, reflecting the presence of naive B cells with no prior exposure to the antigen. Following vaccination, antigen recognition triggers B cell activation, clonal expansion, and differentiation into plasma cells, leading to a marked, at least fourfold, increase in antibody titers. This increase confirms an intact humoral immune response and the formation of immunological memory, ensuring a rapid and effective antibody response upon future exposure to the same pathogen. Similar tests are also conducted to assess T cell proliferation. Engagement of the T-cell receptor with co-stimulatory signals from anti-CD3, CD28 beads delivers the activation signal, switching the T-cell from a resting state to rapid division. Once activated, the cell undergoes clonal expansion, doubling with each round of division. T-cells prove their strength more through two functional tests. The left panel illustrates the delayed type hypersensitivity skin test, which assesses the function of CD4 T helper 1 cells. The right panel shows the chromium release assay, which measures the direct killing power, or cytotoxicity, of CD8 cytotoxic T cells. The delayed type hypersensitivity skin test is a stepwise T-cell mediated immune response over time. Immediately after intradermal injection of the antigen, such as the purified protein derivative used in the tuberculin test, there is little to see beyond a minor local reaction at the site. Over the next 24 hours, previously sensitized T helper 1 cells recognize the antigen and become activated, releasing cytokines, most notably interferon gamma, that act as an alarm signal within the tissue. By 48 to 72 hours, these cytokines recruit and activate macrophages, leading to localized inflammation, tissue edema, and the characteristic induration that is measured clinically. The presence and size of this indurated lesion reflect intact cell-mediated immunity and confirm prior sensitization to the antigen. The chromium release assay is a classic method for measuring T-cell cytotoxicity. First, target cells are labeled with radioactive chromium, which becomes trapped inside intact cells. When cytotoxic T cells recognize and attack these targets, they deliver lethal hits using perforin and granzymes, leading to target cell membrane damage and lysis. As the target cells break apart, the radioactive chromium is released into the surrounding supernatant, where it can be quantitatively measured. The amount of chromium detected directly reflects the extent of target cell killing. The greater the chromium release, the stronger the cytotoxic activity of the CD8 T cells. Now let's proceed to the assessment of phagocytes, focusing on two important functions: chemotaxis and intracellular killing. We'll start with the Boyden chamber assay, a classic method for assessing phagocyte chemotaxis. Phagocytes are placed in the upper chamber, separated from the lower chamber by a porous membrane, while a chemoattractant in the lower chamber creates a concentration gradient. Sensing this chemical signal, responsive cells actively migrate through the membrane pores, moving downward toward the source of attraction. Over time, cells that successfully traverse the barrier accumulate in the lower chamber, where they can be counted or analyzed. The number of migrated cells directly reflects the cell's chemotactic ability, providing a functional measure of immune cell motility and responsiveness to chemotactic signals. The nitroblue tetrazolium test, or NBT test, is a functional screen of the neutrophil oxidative burst, in which normal activation of NADPH oxidase leads to the reduction of NBT to dark blue formazine crystals, confirming effective intracellular killing. In contrast, in conditions such as chronic granulomatous disease, defective NADPH oxidase activity prevents the generation of reactive oxygen species, leaving the NBT dye colorless. Today, this functional assessment is more precisely performed using the dihydrorhodamine test, or DHR test, a flow cytometry-based assay in which oxidation of DHR to fluorescent rhodamine directly reflects oxidative burst activity. In normal neutrophils, stimulation activates NADPH oxidase, leading to the production of reactive oxygen species, particularly hydrogen peroxide, which oxidizes non-fluorescent DHR into brightly fluorescent rhodamine 123. This fluorescence is then quantified by flow cytometry, yielding a strong, right-shifted peak. In contrast, neutrophils from patients with chronic granulomatous disease have defective NADPH oxidase, fail to generate hydrogen peroxide, and therefore do not convert DHR into its fluorescent form, resulting in a dull, low fluorescence signal. The clear separation between normal and chronic granulomatous disease fluorescence patterns makes the DHR assay a sensitive, quantitative tool for diagnosing the disease and identifying carrier states. Finally, we enter the complement arena. The complement system is a cascade of proteins that helps clear pathogens. We test the integrity of these pathways by determining whether they can lyse red blood cells. The CH50 assay measures the classical pathway, and the AH50 assay measures the alternative pathway. The CH50 assay is a functional test of the classical complement pathway. Sheep red blood cells are first coated with specific antibodies, which activate the classical pathway when patient serum is added. The C1 complex then binds to these antibodies and sequentially activates downstream complement components, including C4 and C2, thereby forming the membrane attack complex, or MAC. Assembly of the MAC creates pores in the red cell membrane, leading to cell lysis and the release of hemoglobin. The CH50 value represents the amount of serum required to cause 50% hemolysis, providing an integrated measure of the integrity and functional activity of all components of the classical complement pathway. In contrast, the AH50 assay is a functional test of the alternative complement pathway. In this assay, rabbit red blood cells directly activate the alternative pathway without the need for antibodies. C3 undergoes spontaneous activation, allowing C3b to bind to the red cell surface, where it associates with factor B and is cleaved by factor D to form the alternative pathway C3 convertase, a process stabilized by properdin. The reaction requires magnesium, while calcium is chelated with ethylene glycol tetraacetic acid, or EGTA, to block the classical and lectin pathways, ensuring pathway specificity. Formation of the downstream membrane attack complex leads to red cell lysis, and the AH50 value represents the amount of serum required to produce 50% hemolysis, providing an integrated measure of the integrity of the alternative complement pathway. Similar to the CH50 assay, a lower AH50 result indicates lower functional activity of the alternative complement pathway. And this brings us to the immune report card. When all our tests, from cell counts and identity checks to functional assays, align, we can confidently stamp the report, immune system functional. When numbers, identity, and function are all in order, the immune defenders are fully ready to protect the host.